Okay, so if you're just joining us, next week will be our lecture exam 3. I've highlighted it here. So that'll be on October 20th. Remember, we're just a week behind. So come to class on Tuesday for an exam on chapters 7 to 9. And like everything else, I'll post the Google link in the chat. If you have the lab exam today, I'll post the Google link in the chat in the Zoom lab link for the exam at 1 p.m. today for the lab exam. Just keep in mind, next week, we're probably going to make up these 2 labs in one. It still shouldn't be too bad because labs don't take that long, but just keep that in mind for lab next week. You'll probably have 2 lab activities to do. So if you can't make it or there's some sort of extenuating circumstance, let me know because there will be probably 10 points given out in lab next week. And today we're just going to do chapter 9, which will be the senses. And then again on Tuesday, you will have a lecture exam on these chapters. So this is chapter 9, special senses. And again, if you guys have any questions, just put them in the chat here. So we kind of are finishing out our nervous system talk. And we always end in anatomy textbooks by talking about your senses because they have to do with your nervous system, but they're kind of separate from the other nerves, brain, and spinal cord. So a sense is the ability to perceive any sort of stimuli, whether it's an external or an internal factor. And a sensation is the conscious awareness of the stimuli received by your sensory neurons. And sensory receptors are throughout the body. They are sensory nerve endings that respond to any sort of stimuli by developing an action potential. And
no. Okay, no big deal. Okay, cool. Go ahead whenever you're ready. Sorry about that.
And
good morning, everyone. My name is Maisha.
I got to figure out how to mute people. Sorry, I haven't been on Zoom in a while. There we go.
Physical tension. It usually makes.
There we go. Okay. An action potential, like we've talked about before, is anytime a neuron fires or gets electrified. Think about it like that. So here's our classifications of senses. They can be a general sense. So these receptors are distributed all over the body, like in your skin, your muscles, your joints. Those are the somatic senses. And then visceral senses are located in internal organs. And then your special senses are smell, taste, sight, hearing, and balance. And we'll go over some of the special senses today too. So your general senses, again, these receptors are over large parts of the body. Usually these senses involve feeling touch, pressure, pain, temperature, itch. Somatic provides information about the body and the environment. And visceral, remember that word visceral always has to do with viscera, your internal organs. And then these are your special senses. Different types of receptors. So again, a receptor is any sort of molecule that can detect a stimulus. A lot of them are located in your skin, but they can also be located internally, detecting some sort of stimulus in your internal organs. Mechanoreceptors detect movement, and you should know the difference between these types of receptors. So mechanoreceptors detect movement, so these will detect touch, pressure, vibration. Chemoreceptors will detect chemicals like odors in your nose. Photoreceptors are found in the back of your eye. They detect light. Thermoreceptors will detect temperature changes, and then nociceptors will detect pain. So those are the 5 different types of receptors. We divide the touch receptors. There's several different types of touch receptors, and many of these are found in your skin. The Merkel's disc will detect light touch and pressure. The hair follicle receptors also detect light touch. Meissner's corpuscles are located deep in your epidermis, and they will be where other touch sensations or tactile sensations are located. Merkel's disc and Meissner's corpuscles are probably named after the people who found them or discovered them first. Just like Ruffini corpuscles and Pacinian corpuscles, Ruffini corpuscles are deep tactile receptors. So they will detect continuous pressure located deeper in the skin. And Pacinian corpuscle is the deepest skin receptor. They are associated with your tendons, your joints, and they will detect deeper pressure still, vibrations, and position, especially having to do with the tendons that surround your bones. So a lot of this is kind of just basic memory work about what these different touch receptors are. So you can see where the different touch receptors are and where they're located kind of in different layers in the skin. Here's your Meissner's corpuscles and what they do. Ruffini corpuscles, Pacinian corpuscles are the deepest or most deep, so deepest further into the skin. You have hair follicle receptors surrounding the hair, and then Merkel discs are the most superficial, detecting light touch. You also have just free nerve endings, which will respond to any sort of pain sensation, change in temperature, itch, or movement. So those are the different sensory receptors. Pain is an unpleasant perceptual and emotional experience. Don't you just love that definition? Unpleasant. Pain can be localized or it can diffuse away from its point of probably sensation. The localized will be sharp, pricking, cutting pain. This will include with rapid action potentials. A diffuse pain is characterized by a burning or aching pain, and its action potentials will be slower or longer lasting. Pain control, we have two types of anesthesia, local or general. Local anesthesia, the action potentials will be suppressed from that pain exactly where it's located. So the receptors will be in local areas. The chemicals will be injected near the sensory nerve and be able to kind of take out those pain sensations that sensory nerve is taking up to your brain. General anesthesia is what we characterize as a loss of consciousness and kind of going under. And these chemicals will affect the reticular formation in the brain. Referred pain is very important, especially if you work in an ER. Referred pain originates in a region that is not the source of the pain stimulus. It will be felt when internal organs are damaged or inflamed, and the sensory neurons from the superficial area and neurons from the source of the pain will converge into the same ascending neurons in the spinal cord. So basically, the same sensory neurons from that area felt in the body will kind of travel the same pathway up to the brain in the same spinal cord segment as the same sensory neurons coming from that particular internal organ. So the biggest example of this is what's one characteristic of a heart attack. If you have a problem with your heart and a part of your heart is damaged, where will you find or perceive that pain from?
Arm pain.
Know?
Arm,
the arm.
Your arm, yeah.
Specifically the left arm.
Unless you're a female, and most commonly female, it's either abdominal pain or leg pain.
Also down there. Yes, thanks, Tristan. So these are just examples of referred pain. So pain in the heart, the same sensory receptors coming from the left arm would travel that same pathway up to the brain as the same sensory receptors coming off of the heart. So we call this referred pain. It's being referred to another part of the body. Another one is the appendix. So sometimes the appendix, it can be referred to, or the colon can be referred to around your belly button. So again, this is important if someone presents to you in the emergency room, or even a family member presents to you with extreme pain on their left arm, they could be having a heart attack. The liver and gallbladder are way up here in your right shoulder. So kind of interesting, referred pain. Olfaction is the sense of smell. So we'll start to get into your special senses here. It occurs in response to odorants, which will travel in through the nasal cavity. Receptors will be located in the nasal cavity and the hard palate. So here's the hard palate. It's the top of the roof of your mouth. And then you have fibers of the olfactory nerve coming through a cribriform plate detecting any odorants. And we can detect 10,000 different smells. Isn't that crazy? I can't even think of 10 different smells right now. Well, I probably could. So lots of different odorants. The olfaction process, the nasal cavity contains a thin film of mucus where the odors will dissolve. The olfactory neurons will be located within that mucus, and dendrites of the olfactory neurons will be enlarged and contain cilia. The dendrites pick up the odor, depolarize, that just means that they're being electrically stimulated, and will carry that odor to axons in the olfactory bulb, which is cranial nerve number 1. Your frontal and your temporal lobes of the brain process the odor. So your the olfactory nerves will take in that kind of stimulus to your brain, and then your brain figures out what you're smelling in your frontal and your temporal lobe. So this is just a look about olfactory epithelium and the olfactory kind of sense of smell. So here are your olfactory neurons with the cilia detecting those odorants, and then it'll travel up through this olfactory neuron and axon, the olfactory nerves, and eventually synapse on the olfactory bulb, which will take it to the brain. Taste, this is often called gustation. So your taste buds are sensory structures that detect taste. They're located on papillae on tongue, and the papillae are all the bumps on your tongue. So some people think that the bumps that you feel on your tongue are the taste buds. Those bumps are called papillae, and the taste buds are within those papillae. You have papillae on your tongue, your hard palate, also in your throat. And inside each taste bud are 40 different taste cells. Each taste cell has different taste hairs that extend into the taste pores. So here's a look at all the bumps or the papillae. You can see the papillae is right here, and within kind of the depressions between the papillae, you have all these taste buds. And you can see what a taste bud looks like here with the taste cells, the taste hairs. And again, they're all connected to some sort of neuron at the bottom that'll change that chemical, the taste, into an electrical signal that'll go to your brain. So then your brain can figure out what you're tasting. So how do we taste? Your taste buds will pick up the taste and send it to your taste cells. The taste cells send the taste to the taste hairs, and the taste hairs contain receptors that will initiate an action potential. Again, some sort of it'll electrify that nerve, which will be carried to the parietal lobe. So your taste will be sensed in the parietal lobe, and that is where your brain will process the taste and determine which type of taste your tongue has picked up, whether it's sweet, sour, salty, bitter. Umami has to do with the breakdown of protein. Certain taste buds will be more sensitive to certain tastes depending on where they're located on the tongue. And taste is also linked to smell. So I don't know if you guys have ever gone wine tasting, but they usually have you smell the wine and then you drink it. So taste and smell are closely linked. I've been wine tasting like once. I should probably go more often now that I live in California, but I think they're all closed. Are wineries still closed? I don't know.
I don't think
they're
so.
open. No? Okay. Let me know. If any are open, let me know. Pathways for the senses taste. So this just shows how different papillae are located in different parts of the tongue and throat, and then they'll travel up through different nerves. These refer to different cranial nerves, and then up to the parietal lobe in the brain. Again, I don't know if this was the lecture I recorded or what we talked about, but all sensory information usually goes through the thalamus first. The thalamus takes in all sensory information from all parts of your body and then projects it onto the correct lobe of the brain that needs to process it. Then we get to vision, and we'll talk a lot about vision kind of anatomically, some of the anatomical structures. And I think in lab next week, I'm sure you guys will be going over some of these structures in lab. So you'll have to know these accessory structures of vision. Your eyebrows are important for protecting your eyes from sweat, also shading from the sun. The eyelid and eyelashes protect from foreign objects. They also lubricate the eye by blinking. So your eye is constantly surrounded by the tear or the watery substance, which your glands make. And the watery substance, your tears, contains enzymes which help to keep your eyes clean and also protect them from anything foreign. And your eyelids, the blinking just helps to move that fluid around the exterior part of the eye. So here are the accessory structures of the eye, the eyebrow, eyelashes, eyelids, you guys know these. Some other things, so the pupil is the black part of the eye, and the iris is the colored part that surrounds the pupil. The iris is actually made up of two different types of smooth muscle that helps to control how your pupil dilates, gets larger, and constricts, gets smaller. Conjunctiva, so these are some more kind of external features you can see. The conjunctiva is the thin membrane that covers the inner surface of the eyelid. The lacrimal apparatus will be kind of this whole structure that will produce tears and drain tears. And then you have 6 extrinsic eye muscles that help move your eyeball. So here's a look at the lacrimal gland and the lacrimal apparatus. So all of your tears are formed in the lacrimal gland, and they're released kind of on the lateral upper side of each eye through lacrimal ducts. So the direction of tears always flows from the upper lateral side of your eye across the eye to this medial lacrimal canaliculi, which are little tiny ducts that drain the tears in the medial side. Then the tears will travel down the lacrimal sac and empty into the nasolacrimal duct. Usually all of your tears, are used up with the eye and evaporate. Not many will drain into the nasolacrimal duct. But if you're really sad and you start to bawl and start crying, that's why your nose kind of drips a little bit, because all these excess tears are being drained into the nose through this nasolacrimal duct. But tears are important for keeping your eye clean.
Can you go back?
Yeah.
To one more.
Yeah.
Okay, I just wanted to see those words one more time. Thank you.
Just responding to one message here in the chat. Okay. All right, so here are the extrinsic eye muscles. I teach anatomy several different places. We talked about these, I think, the 6 eye muscles.
We did.
We did, thanks. Okay, so yeah, you have 6 eye muscles attached to the top, bottom, medial, and lateral sides, and then you have 2 oblique muscles, superior and inferior, that kind of attach to the eyeball at an angle. So 6 different muscles for your little tiny eyes, and you'll notice because you can move your eyes in a million different directions, and those are all the function of these muscles. The anatomy of the eye then, in general, it's a hollow fluid-filled sphere, and it's composed of 3 different layers, or tunics we call them, and we divide the inside into an anterior and a posterior chamber. So if we look at the eye, and again, we'll cover this in lab next week, I believe. If we start at the front of the eye, you have a cornea, which is the outer part of the eye. This is known as the fibrous tunic, made up of the sclera and the cornea. The sclera are the whites of the eyes, and the cornea is transparent, and it's the place where you would put probably a contact lens. So that's the fibrous tunic. The vascular tunic is the second layer, and that's made of the choroid layer, which is the second layer. These are where all of your blood vessels will be found. Also, the vascular tunic has the ciliary body, which controls the suspensory ligaments that will control the shape of the lens, which is this structure. The lens is this structure that will control where to focus the light as it comes through the eye. And these suspensory ligaments, they kind of look like little strings, are attached to the lens, and the ciliary body is a special muscle that controls how those suspensory ligaments control the shape of the lens. Here is the pupil. You can't really see it from this angle, but then the iris will control the shape of the pupil. The anterior and posterior chamber have to do with kind of in front of the iris and pupil and behind the iris and pupil. The vitreous chamber is filled with vitreous humor. So everything, the anterior and posterior chamber, which are in front of the lens, are filled with more of a liquid, watery type substance. The vitreous chamber, which is all this back here, that's filled with more of what we call vitreous humor, and that's like jelly-like fluid. And that just helps to keep your eyeball, keep its nice spherical shape. The retina is called the nervous tunic, because that's where all of your nerves and photoreceptors will be. The optic nerve attaches to the back of the eye right there. So this describes the fibrous tunic. This is your outermost layer. Your sclera are the white part of the eye, and then the cornea is the transparent structure that covers the iris and the pupil. It will allow enter and focus the light, and the sclera helps to maintain your eye shape and provides attachment sites for your muscles. The vascular tunic is the middle layer, contains all your blood vessels. The choroid is the black part that contains melanin, and it will deliver oxygen and nutrients to the retina because it will contain your blood vessels. The ciliary body and suspensory ligaments help hold your lens in place. Vascular tunic contains the lens, which is a flexible disc. It will focus light onto the retina, which is the back of the eye. The iris is the colored part of the vascular tunic. It'll surround and regulate pupil size because the iris is actually a smooth muscle. The pupil then regulates the amount of light entering the eye. If you guys are in a really sunny area, like if you would walk outside today, your pupil would get really constricted. And if you're in a dark room, your pupil will become dilated. If it becomes dilated, it's trying to take in more light. And if it becomes constricted, it means there's a lot of light there and it's trying to kind of keep out the light from entering. So here's a look at how the lens and the ciliary body are connected. So here are the ciliary muscles of the ciliary body, and they have little suspensory ligaments that come out from them to connect and touch the lens. So this is showing the lens and the ciliary body. The ciliary body are muscles connected to suspensory ligaments that will control the shape of the lens. The iris then controls the shape of the pupil, and it's made up of circular smooth muscles that surround kind of in a circle the pupil, and radial smooth muscles come out from the pupil kind of that way. So just two types of smooth muscle are in the iris, and they will control how your pupil will dilate or constrict. The nervous tunic is also known as the retina. This is the innermost tunic. The retina covers the posterior 5/6 of the eye. So most of the eye, the back of the eye is covered in retina. It contains two layers. The pigmented retina is the outer layer. This keeps light from reflecting back in the eye. And the sensory retina is the layer that contains all of your photoreceptors. Your photoreceptors are what will be stimulated. These will contain your interneurons. So when light enters the eye, it'll be focused on your photoreceptors, which will take that light sensation to your brain. You have two different types of photoreceptors, rods and cones. Your rods are photoreceptors that are sensitive to light. There are 20 times more rods than cones in your eye, and your rods can function in extremely dim light. So when you walk into a dark room, you'll notice it takes about 20 minutes for your, maybe less than that, 10 to 20 minutes for your eyes to adjust to the light. And that's because your rods are trying to take in as much light as possible. So after 15 minutes, it's a lot easier to see in a dark room. And that's because your rods have kind of adjusted to the dim light. The cones are the other type of photoreceptor. They are the photoreceptor that provides color vision. So there's three types of cones. You have blue, green, and red types of cones. And people who are color blind could have some sort of defect in one or all types of cones, because some people are completely colorblind. Others are just colorblind to different colors. So depending on which type of cone has the problem. This is what a rod looks like. And this is what show you what a rod looks like and what a cone looks like. That's wrong. That should say cone up at the top. So rod just looks more like a rod in its outer segment. And basically this is just the photoreceptor. So light will hit this rod and be connected to some sort of neuron to take that information up to the brain. And then the cone looks more cone shaped at the top. And again, it's just the photoreceptor that detects different colors. Daisy, you're colorblind. We told that to everyone, so it's not private. But yeah, we'll talk about being colorblind in a little bit. And I'll test you guys here. I have a slide that tests you. Some pigments and pigment proteins. Rhodopsin is the photosensitive pigment in rod cells. Opsin is the colorless protein in rhodopsin. And retinal is a protein pigment. It's the yellow pigment in rhodopsin, and it requires vitamin A. So these just are different pigments and proteins that are important for detecting light and color in these photoreceptors. So the effects of light on rhodopsin. So how does rhodopsin get affected by light? Well, the light will strike the rod cell. The retinal will change shape within the rod cell. Opsin will change its shape. The retinal will dissociate from the opsin, and it'll change the rhodopsin, this protein pigment shape. And the change in that rhodopsin will stimulate the response in your rod cell, which will result in vision. The retinal will detach from the opsin, and again, these are just different proteins. ATP is always required to reattach the retinal to the opsin and return the rhodopsin to the original shape. So these are just the effects of light on this rhodopsin, this color protein pigment in your rod cell. This takes you through kind of what I just talked about. Rhodopsin is composed, so here's the rhodopsin pigment. It's composed of opsin and retinal. Light causes the retinal to change shape, which activates the rhodopsin. The activated rhodopsin will stimulate the cell change that results in vision. Following rhodopsin activation, the retinal will detach from the opsin, and energy will be required to basically move the retinal back into its original shape and the original form of the rhodopsin pigment. So you kind of get this cyclical process of light touching this rhodopsin pigment, the retinal detaching, and then energy is required to put the retinal back into this protein rhodopsin. So this is kind of nitty-gritty how light affects rhodopsin in the rod photoreceptor. The retina so the retina again is the third, it's the innermost layer of the eye. This is where your rods and your cones will be. They will synapse or join with bipolar cells of the sensory retina. Horizontal cells of the retina modify the output of the rods and cones. These bipolar and horizontal cells will synapse or pass along their electrical signal with ganglion cells, and ganglion cells have axons that converge to form the optic nerve. So these rods and cones take in all the light, all the sensations that you take in through your eye, and it gets passed through several different types of cells to the ganglion cells, which will form the optic nerve. And the optic nerve takes that information to the brain. So here we are in the eye, in the back of the eye, in the retina. And again, we're trying to get all this sort of light sensation to the optic nerve. The direction of light actually hits, so we travel all the way to the back of the eye first, where the pigmented retina and the photoreceptor layer is. So light travels all the way back to the photoreceptor layer, and then the direction of the action potentials will be propagated back kind of towards the front of the retina layer. So we have your rods and cones back here, and they will be stimulated and pass along an action potential to all of these interneurons, which are made of horizontal and bipolar cells. Those will eventually synapse on ganglia cells, which have axons that will create the nerve fiber to the optic nerve. So this is just the source of how light is picked up, perceived, and then sent on down the optic nerve. Any questions on this? I don't think you need to know like all the details of this. We're just trying to give you a general kind of basic function of how your photoreceptors pick up light and then send it on down to the optic nerve. Within the retina, you have an area called the macula, which is a small spot near the center of the retina. Within the macula, you have a point about the size of the end of a pin. This is the center of the macula, and we call that the fovea centralis. And the fovea centralis is where your light will always be focused when looking directly at an object. So your eyeballs are constantly kind of changing, just changing their direction so that light will be constantly focused on the fovea centralis. Within the fovea centralis, you only have cones there, and cones are responsible for color vision and visual acuity. So within the fovea centralis, this is kind of the area of the retina where you have the highest or the sharpness of vision. Within the fovea, you have the ability to discriminate fine images. Also in the retina, you have the optic disc. This is a white spot directly medial to the macula. This is where your blood vessels will enter the eye and spread over the retina. The axons will exit as the optic nerve. So because you have blood vessels at this optic disc area, this is where your optic nerve attaches to the eye, and you won't have any photoreceptors in this area. This is also called your blind spot. So when you're focusing on something in your peripheral vision, eventually if you bring it closer to your eye or further away, it'll enter into this blind spot, we say. But this is the area of no photoreceptors in the optic disc. So if we look at kind of the inside of the eye, this is showing the optic disc where the optic nerve attaches, and you can see all your blood vessels coming out of that area. Right medial to that, we'll see your macula, and within your macula, you have the fovea centralis. And excuse me, I said the optic disc is medial, the macula is more lateral. This is a test for testing your blind spot. If you focus on and you might have to do this when you download this PowerPoint later, but if you focus in on the red dot and cover your right eye and then bring the screen to and from your face within about 5 inches, in your peripheral vision, this plus or cross sign will disappear from vision. And you can try that out on your own or Google blind spot test. Okay, questions so far? All right, we'll continue on. Chambers of the eye. So these are chambers within the eye. The anterior chamber is located between the cornea and the lens. It's filled with watery aqueous humor, which helps to maintain pressure and refracts light. Does anyone know what the word refracts mean?
Take in, I guess, and change it.
Close, very close. It just means to bend the light. So changing it, bending it. The aqueous humor also provides nutrients to the inner surface of the eye. The posterior chamber is just located behind the anterior chamber, and it still contains that aqueous, watery humor. The vitreous chamber is then located in the retinal region. It'll be located behind the lens. It's filled with this jelly-like substance that helps to maintain pressure, hold the lens and retina in place, and also works to bend or refract the light. The functions in the eye, it functions much like a camera. The iris allows light into the eye, which is focused on the cornea, lens, and humors onto the retina. And the light striking the retina produces action potentials, and some of those cells we talked about that are then relayed to the brain. Light refraction, which is light bending, and image focusing are two important processes in establishing vision. So light refraction is the bending of light. The focal point is the point where the light rays will converge in the back of the retina. It'll occur anterior to the retina, so it'll invert the object. And I hope they show you a picture of this. They kind of do. Okay. Focusing images on the retina. Accommodation is where the lens will become less rounded and the image can be focused on the retina. So basically the light coming into your eyes, the shape of the lens determines how the image is focused on the retina. If the lens becomes less rounded, it enables the eye to focus on images closer than 20 feet. And this just shows you how your lens changes shape, whether you're looking at something very distant, further away, versus something closer or near. And you'll notice here the FP stands for the focal point. So the lens will merge the image to the focal point and then invert it to the back of the eye. So all of the images that you're seeing right now are actually inverted. They're upside down on the back of the eye. Your brain is what puts them right side up so that you can see everything right side up. So if we're looking at something distant or further away, the lens becomes flat due to the work of your ciliary muscles and the suspensory ligaments. So your lens becomes flat to look at something further away. And to look at something closer, your lens becomes thickened. It's harder as you get older, for the lens to accommodate images that are close up. So it's harder for your lens to become thickened. And that's why many people have to wear reading glasses to focus on things like print that are closer to the eye. Okay. Are you guys still there? My screen just stopped.
We're here.
Here, sorry.
Yep, still here.
Thanks, my PowerPoint just, we'll try this again. Okay,
I'm still here.
thanks. Glad you guys are here. All right, neural pathway for vision. This just means the pathway for neurons. The optic nerve, so after your photoreceptors sense the light in the images, how do those images get to your brain? They travel through the optic nerve into the optic chiasm, where your two optic nerves like crisscross each other in an X shape. And optic tracts are the route of those ganglion axons that bring the sensations to that part of the brain. So this is showing the optic nerve has been cut here, but this will go to both eyeballs. And the optic chiasm will be where your optic nerves crisscross each other, and they'll continue on as the optic tract into the thalamus. And then the thalamus will send out the sensation to the visual cortex located in the occipital lobe. So this shows the visual pathway. This gets a little confusing. Each visual field is divided into temporal and nasal halves, meaning one on the lateral side and one on the medial side. And everything you see that enters the nasal field or the medial side will be projected to kind of the left side of your eyeball. And really simply put, what this is all saying is, since your optic kind of area is tracked, everything that you see from the left side of the body will go to the right side of the brain. And everything that you take in on the right eyeball will go to the left side of the brain. So that's what this really complicated kind of 6 steps is showing, how the visual fields take in vision and images and then project them to that area of the brain. Visual defects. So myopia is nearsightedness. This is what most of us have who wear glasses right now. We are unable to see things far away. That's being nearsighted. The image is focused in front of the retina. Hyperopia is farsightedness. That's you're unable to see things close up. The image is focused behind the retina. Presbyopia is where the lens becomes less elastic or has less ability to accommodate. So those are visual defects. Astigmatism is where you have an irregular curvature of the lens. You usually have to wear corrective contacts or glasses to correct this. Astigmatism, if you're looking at a clock or any sort of circle with radial arms coming out of it, some of the clock arms or some of the numbers in the clock would appear blurry or more bolded than the others. So that's what astigmatism would look like. Color blindness has the absence or deficient cones. Primarily it affects males, but not always. And again, depending on what cones are defected, you could be colorblind to only different colors. I'm sure you guys have seen those corrective, they're so popular on social media. You watch those videos of people getting the corrective glasses and they're able to see colors, which is so heartwarming, but I think they're actually really expensive. But that would correct some of these cones that are deficient. Do you guys know what I'm talking about? Maybe I only get those videos because I'm a mom and they know I'm more emotional.
Yeah, those glasses are around almost $500.
Oh, they are expensive. Okay. Glaucoma is increased pressure within the eye. So that's what glaucoma is. Glaucoma causes the vision to be kind of blurry around the edges, kind of like astigmatism, but due to more pressure buildup. Okay, so here you can test for color blindness in yourselves. Hopefully, if you were colorblind, you wouldn't be able to see a number pop out at you because there's only the colors are made of circles. So what do you guys see in letter A? Do you see a number pop out at you?
Okay, I do. I see 74.
What about in B?
5.
Okay. Did you say you were colorblind, Daisy?
Yeah, and I see it good. This one I see good. Maybe I'm not colorblind. I don't know my situation, but It's weird between green and blue. It's
Green and blues. Okay,
Yeah.
and these are more, so this is really testing green and blues versus reds and oranges. So you probably wouldn't show you. You would have to look at a green and blue test.
I mean, it also could be different being that she is over the computer and the way the chromatics were over computer compared to paper.
Yes. So if we were in lab, I've taught some labs where there's like colorblind books you can buy and go through the book and test that. Oh, Ryan, thanks for, I'm just reading you're in the chat. Wow, so she was 27. Yeah, so usually some people can be slight colorblind and they don't really know it because it could be just off very basic, like you said, like a Ryan versus, or like a brown versus a burgundy. Anyone else colorblind? You don't have to share, but I've had students who found out they were colorblind in class after working through this like book, which is, I mean, you can totally live a normal life, but some of the colors might be a little off. Want to move on to the ear? Everyone with me?
Yes, very interesting though. Very interesting.
Brian, my wife says I can't match my clothes, so I might be. I don't know. She could be joking, but yeah, you could be. Is she serious?
Yeah, serious, super serious.
Okay. Well, yeah, get yourself a colorblind book. You can test it or go to the eye doctor.
She's probably messing with him, saying he didn't like what he's wearing.
Could be. Oh, that's funny. All right, moving on to the ear. So the ear, the organs of hearing and balance are located within the ear. So your ear isn't just about hearing, it's also how your body maintains its balance and its perception of balance and equilibrium. We divide the ear into 3 areas: external, middle, and inner. The external ear is made up of your auricle, which is this fleshy part we all have different shapes of. That's your auricle, the external ear. The external auditory meatus is the canal that will lead to the eardrum. The tympanic membrane is another word for the eardrum, and that will separate this external from the middle ear. And the tympanic membrane is a really thin membrane that separates those 2 chambers. And the middle ear is filled with air. So that's something to remember, that the middle ear is not filled with fluid like the inner ear will be. It's made up of 3 tiny ossicles. These are the tiniest bones in the body. I think I talked about during our skeleton talk is they will all fit on the head of a penny. The malleus, incus, and stapes. The malleus is directly attached to the tympanic membrane. The incus will connect the malleus to the stapes, and the stapes looks like a stirrup that you would ride if you were on like a saddle of a horse. And this is the bone located at the base of the oval window, which we'll talk about. The oval window is kind of another membrane type structure that separates your middle and your inner ear. And then the eustachian, also known as auditory tube, opens into your pharynx or your throat. And eventually I'll get to a picture here. This will equalize air pressure between the outside air and the middle ear. So when you're going up into the mountains or in an airplane, your ears will pop. And that's the work of your auditory tube, trying to equalize pressure between the outside air and the middle ear. The inner ear then is all filled with fluid. There's 2 kind of chambers, the bony labyrinth and the membranous labyrinth. The bony labyrinth are tunnels filled with fluid. This is where your cochlea, your vestibule, your semicircular canals are located. They are filled with fluid. The membranous labyrinth is located within that bony labyrinth, and it's filled with a special type of fluid called the endolymph. Where's the picture? Oh man, finally. Well, just listen to me talk and then we'll get to a picture. The endolymph is the clear fluid within the membranous labyrinth, and the perilymph is the fluid between the membranous and the bony labyrinth. So just different types of fluid within these inner ear chambers. The cochlea looks like a snail, and the cochlea snail shell is where hearing takes place. Within the cochlea, we have different kind of channels or canals. The scala vestibuli is filled with perilymph. The scala tympani is in the cochlea, filled with perilymph as well. So they're just kind of 2 different kind of inner channels within the cochlea. And there's a cochlear duct between the two that's filled with endolymph. And I'll show you a picture eventually. Thanks for bearing with me. The spiral organ is within the cochlear duct. The spiral organ is where your hair cells will be. The tectorial membrane is in the cochlea. Basically, you have little tiny hair cells that will be embedded in this tectorial membrane, and the hair cells will bend when sound vibrations will pass through this fluid, and the bending of those hair cells produce action potentials that will be sent up to your brain. The vestibular and basilar membrane are different walls that separate out the scala vestibuli from the scala tympani. Let's get to some pictures. Okay, so here's the structure of the ear. Here's your auricle right here. So your auricle kind of looks like its purpose is to funnel all sounds in through this external auditory canal. The external auditory canal ends at this tympanic membrane, and that's kind of the boundary between your external and your middle ear. The middle ear is filled with air, and it has these 3 tiny bones, are called ossicles. The malleus is connected to your tympanic membrane. The malleus is connected to the incus, and the incus is connected to the stapes. So what happens with sound? Sound waves travel through the external auditory canal, and they vibrate the tympanic membrane, your eardrum. Those vibrations get sent through these teeny tiny bones. And because the sound waves are so much larger than these ossicles, those vibrations are amplified at least 100 times through the ossicles. So basically in sound, so what your ossicles are doing right now, they're hearing my voice, your ossicles are sending out vibrations. Those vibrations kind of do this, they cause the stapes to go like this against the oval window. So those vibrations get sent out through the oval window, and the pounding of the stapes on the oval window will send these sound waves or vibrations through the cochlea. And remember, within the inner ear, these are all filled with liquid. So this liquid now carries the sound waves traveling through the liquid. And within the cochlea, you have hair cells. And when those sound waves travel through the liquid, they'll cause your hair cells to bend. And the bending of those teeny tiny hair cells cause action potentials to travel through your cochlear nerve. The semicircular canals are where kind of your balance and equilibrium takes place. So the vestibulocochlear nerve is one of your cranial nerves, and it has a vestibular branch that goes to the vestibule and the semicircular canals, and the cochlear branch that goes to your cochlea. Okay, so the structure of the inner ear, so this is more of a look. Here's your semicircular canals. The vestibule is the area right kind of underneath your semicircular canals. And here is your cochlea shaped like a snail. So your cochlea, if we unravel the cochlea, so it looks like a snail shell, but if we unravel it and then we kind of take a cross section of it, you'll see that the unraveling kind of within that cochlea, you have these 3 different chambers. And these 3 different chambers, you have a scala vestibuli at the top, the scala tympani at the bottom. The important part that I want you guys to focus on is this, it's called the spiral organ, or the organ of Corti. And it's zoomed in right here. And within here, you can kind of see these hair cells. The hair cells are embedded in this tectorial membrane. And really simply put, these sound vibrations travel through the cochlea, and they cause these hair cells to bend. And the bending is picked up on the axons of this cochlear nerve. And that bending of the hair cells is what your brain perceives as sound. Good with that? Kind of? Okay. So the hearing process, this kind of takes you through what I just talked about. The sound travels in waves through the air. It vibrates your tympanic membrane. The sound is amplified by those ossicles, which will transmit those vibrations to the oval window. The oval window produces waves in the perilymph, which is just the fluid of the cochlea. The vibrations cause the vestibular membrane and the endolymph to vibrate, Those are those inner chambers of the cochlea, and the endolymph is the fluid within that inner chamber. That causes displacement of the basilar membrane, which is where your hair cells are located. The movement of the basilar membrane is detected by the hair cells in the spiral organ. When the hair cells bend, they cause an action potential to travel through the neuron of that cochlear nerve. So here's a look at the cochlea, but it's been unraveled a little bit to show how sound travels from 1 to 2 in the middle ear to 3 at the oval window. Those vibrations now are becoming waves through the fluid, and they'll travel through the cochlea, and they'll eventually those vibrations will exit out of the round window, which will kind of lead them back to the middle ear. The auditory tube is also shown here. Again, the auditory tube helps to maintain pressure with the external air. And this is where you might hear a popping sound due to the popping of the tympanic membrane, popping it back into place from any sort of change in pressure. Balance and equilibrium. This will have to do with your vestibule and the semicircular canals in the inner ear. Static equilibrium is associated with your vestibule, and it will evaluate the position of your head relative to gravity. And dynamic equilibrium is associated with these semicircular canals, and this will evaluate change in direction and the rate of head movement if you're moving your head back and forth really fast. So the vestibule is your inner ear. It contains what we call the utricle and saccule. The maculae are specialized patches which contain hair cells that will again pick up any sort of change in balance within the vestibule. The otoliths are a gelatinous substance that will move in response to gravity. They will attach to hair cell microvilli, which will initiate action potentials, again, in the vestibule. So this is a look at location and structure of of the maculae. And you can kind of see this is your vestibule where your utricle and saccule will be located, showing how the otoliths are located in this gelatinous mass. And these microvilli, these teeny tiny hair cells, will pick up any sort of movement in balance, and then the hair cells will bend, and the bending of those cells will be picked up by these nerve fibers of the vestibular nerve. This just shows the function of the vestibule in maintaining balance. So again, let's just say you're looking straight up. You can see the hair cells are not bending, but if you look down, the hair cells will bend due to the force of gravity, and the bending will be picked up by these vestibular nerve fibers. Balance is then mainly taken up in the semicircular canals. We call that dynamic equilibrium. It senses movement in any direction. The ampulla is the base of the semicircular canals. The crista ampullaris is within the ampulla. And the cupula has to do with a gelatinous mass, again containing microvilli. It will float, but it'll always be displaced if there's any sort of movement or change in balance. So here's a look at the semicircular canals, showing how these hair cells, again, have microvilli. They're embedded in this gelatinous mass, and if there's any sort of change in balance or equilibrium, the hair cells will bend, and that change, the bending of the hair cells, will be taken up and cause an action potential to travel through the vestibular nerve. So I know this is kind of confusing, but just really with any sort of like hearing structures, whether it's sound waves or balance or equilibrium, it's all just bending the hair cells in different parts of the ear. And if you can remember the bending of these hair cells, that is what causes an action potential, or that nerve to send off the electrical stimulus to the brain. Oh, and that is the end of that. So I'll stop the PowerPoint here, stop the recording.